Hello everyone and welcome to ITLS Academy Empower the Youth. So today in this lecture we will going to study about a new experiment, that is to study methylene blue reduction test for given milk sample. So the introduction part of the topic says that quality analysis of milk by SCP and MBRT analyzes the microbial profile present in milk. The analysis thus derived help in deriving the data of bacteriological population of the milk and the relative effect on the quality of milk. MBRT, that is methylene blue reduction test, helps in evaluating the contaminant factor as involved due to the presence of microbes in milk, whereas SCP, that is standard plate count, denotes the number of aerobic microbes present in the milk. So these two tests further help in deriving conclusion regarding the quality of milk and its effect on the health of the consumer. So as we all know that milk is a basic essential for the development of any human body. And if microbes present in the milk, they can contaminate the quality of the milk. When the quality of the milk is contaminated, and if not treated, if that contaminated milk is being consumed by any human, so it can lead to different types of diseases. So it is very much important to treat the milk sample, to treat the milk before the consumption. So we all know that the quality analysis of any milk. So if you see this thing, that how the quality analysis of any milk take place. So if you talk about a tone milk, so it is obtained after combining buffalo milk and cow milk. Okay, so tone milk is obtained after the combination of both the milks, that is buffalo and the cow milk, to attain a concentration of 3% fat and 8.5% that is not fat content in the milk, including the protein and sugar, as compared to 3.5 to 4% dissolved fat in the regular milk. Thus an abstract of the quality analysis of the tone milk. So as per the FSS's regulation and according to the Indian standards, the fat content in the toned milk cannot get lower than 3%. So according to the standards, the fat content should be 3%. So in the toned milk, okay, so if you see that in a toned milk, the significant content of vitamin A and calcium is present in toned milk. So the average, you can say the SCP, that is standard plate count, shows an average plate count of 30,000 grams in the toned milk found in India, and it reflects the quality of the toned milk is comparatively higher than that of a regular milk that are available in the market. So if you talk about the MBRT test, so MBRT test, that is methylene blue reductase test. So it is used to analyze the quality based on the content of the microbiological quality of the milk. So in this type of test, a blue dye, it is added to the milk, and its rate of discoloration defines the microbiological quality of the milk. The sooner the color decolorizes, reflect the inferiority of the microbial quality of the milk. Okay, so if you can see on the screen, there is a diagram, or you can say a figure, that shows a volumetric flask of a methylene blue solution. So why there is a need to study the methylene blue reduction test for the given milk sample? So as we all know that one of the best medium for the growth of microorganism is milk, because milk provide a very favorable condition for the growth of microorganism. So both the raw milk as well as a milk that is pasteurized, they contain varied types of microorganism. So it is because of the oxygen that is released by these active microorganism. What they do, that these microorganism, that the reduction oxidation and reduction potential of the milk get decreased. So some common microorganisms that contaminate the milk are bacillus species, pseudomonas species, staphylococcus aureus. So these are certain types of microorganisms that contaminate the quality of the milk. So this contaminated milk is one of the biggest sources through which diseases are transported from animal to human being, right? So mostly the milk that is contaminated, when it is handled properly during the milking process, the microorganism from the exterior surface of the animals come in contact with the milk. So the what are the other sources? So how the I just wanted to tell that how the milk get contaminated. So due to improper handling, or you can say mishandling, or the if the milk is not properly handled during the milking process, then also there are chances of a microorganism to get an entry inside the milk, okay, through the exterior surface of the animal, and they can get in entry in the milk. And what are the other sources of contamination? So the other sources of contamination are like utensils, like milk can in which the milk is stored. If the milk cans are not properly cleaned, they are not washed properly, they are not properly sanitized, then there are huge chances of microbial contamination. And also the milking machine. So the one of the popular tool for the determination of milk quality or is the development of methylene blue reductase, means this test. So methylene blue reduction test is basically used to determine the quality of the milk sample. So what is the basic aim or object behind this? Let us understand that. So milk has significant low redox potential, which reduces the methylene blue immediately. During the milking, cooling and dumping, the oxidation reduction potential of milk is increased by plus minus 0.3 volume. At this point, the methylene blue remains in an oxidized state. So when the bacterial cells are started to increase their number in the milk, it consume more dissolved oxygen from the milk, because microorganism, that is bacteria, it needs oxygen for their survival, right? So as the result, oxygen get depleted. Then the methylene blue start acting as an electron acceptor instead of oxygen. Why? Because oxygen is start depleting. Why? Because oxygen is used by the bacteria cells. The methylene blue gets reduced due to the decrease of oxidation reduction potential from plus minus 0.06 to 0.1 volume. So as you can see, because of the oxygen get depleted, the bacteria, the methylene blue start acting as a electron acceptor. Why? Because instead of oxygen, so the methylene blue get reduced. Why? What is the reason? Due to the decrease of oxidation reduction potential. So how it decreases from 0.06 to 0.01 volume. So the double bond nitrogen atom of methylene blue dies except one atom of hydrogen. As a result, the dye is converted into a colorless state. So the greater is the number of microorganism in the milk, the greater is the metabolic activity, and the faster is the reduction of methylene blue. So if the number of microorganism is greater in the milk, what is the result? The means what is the effect? So the greater is the metabolic activity, and if the metabolic activity is greater, the methylene blue get reduced very fast. So the principle behind this test is that the quality of milk, it is absorbed by milk is determined by absorbing the color that appears in the milk after the addition of dye like methylene blue. Because as we have already understood that this methylene blue, it does, what does it does? It utilizes, or it act as a electron acceptor. What does they do? They act as a electron acceptor, or you can say, because methylene blue is a redox indicator, means which loses its color when it comes under the effect of lack of oxygen, means when there is a lack of oxygen. And how this lack of oxygen take place? Because bacteria utilizes this oxygen very fast for their survival. Okay, so because this methylene blue is a redox indicator, means it loses its colors, start losing its color, and bacterial metabolism, it causes the decrease of oxygen in the milk due to the formation of reducing substances. So the time taken by the dye to get reduced determine the amount of microbes in the milk and the metabolic reaction in an organism. So I am just writing this thing because sometimes in examination and sometimes in interviews also this question generally arises, that the time taken by, you can note it down somewhere, the time taken by the dye to get reduced. Okay, I'm just writing in a short, get reduced, determines the amount of microbes in the milk and the metabolic reaction in the microorganism. Okay, so what are the material that is required for this experiment? So the first material that is required is methylene blue solution, 1% aqueous. Methylene blue solution look like this. Second is a milk sample. Third is the test tube. Fourth is a test tube stopper. 5 is the pipette, and last is the water bath. So these are the material that is required for carrying out the experiment and in the determination of any bacterial or microbes present in the milk sample. Okay, now further move towards the procedure, how the procedure take place. So the first point says, what you have to do, you have to mix the milk sample thoroughly to distribute the fat uniformly, because milk have fat. So you have to mix it the milk very thoroughly and so that the fat get easily distributed. Then you have to add 10 ml of milk sample in a test tube. Now what you have to do, you have to add 1 ml or 10 ml of milk in the milk sample in the test tube. After adding this, you have to add 1 ml of standard methylene blue solution in the test tube, and what you have to do, you have to invert the test tube to mix it properly. Okay, firstly you are adding milk sample. After addition of milk sample, what you have to add, you have to add 1 ml of methylene blue, and you have to invert the test tube and you have to mix it properly. Then after that, Place the test tube in the water bath. Now you have to take the test tube, you have to place the test tube in the water bath at the temperature of 37 degrees centigrade. So what should be the temperature of the water bath? So the temperature of the water bath should be 37 degrees centigrade, and you have to keep it for 30 minutes. Then what you have to do, you have to cover the bath with a lid. Then after 30 minutes of incubation, you have to observe the sample and check for discoloration. Okay, after 30 minutes you take out the sample and you place the sample and check for the discoloration, and make the subsequent reading at hourly interval thereafter. Means you have to make the subsequent reading after each 1 hour of interval. After each reading, you have to remove the discolorized tubes and then slowly make a complete inversion of the remaining tubes. Then what you have to do, you have to record the reduction in the whole hour between the last inversion and decoloration. For example, if the sample was still blue after, let's say, after 5 hours, but was decolorized white at the 2.5 hours, मतलब 2.5 घंटे के बाद, so the methylene blue reduction time would be recorded as 2 hours. Okay, so decoloration is considered complete when 4 to 5th of the color has disappeared. Okay, so in simple words, if we see that what you have to do, you have to take a test tube, you have to add 10 ml of milk sample, then you have to add 1 ml of methylene blue, then you have to invert the test tube and mix it properly. Okay, and you have to do like 4 to 5 times you have to do this, so that it can be easily mixed. Then you have to keep the test tube in the water bath at 37 degrees centigrade for 30 minutes. Okay, now what you have to do, you have to check for the color discoloration. Means after 30 minutes you have to check. So in this thing, what the result shows, in the laboratory, these type of this is the way the experiment is being performed. So if you see the result, okay, so I'm just telling the result first. So you observe a positive result and negative result. Means how? So if the viable bacteria decolorizes the milk within 30 minutes, means within the starting of 30 minutes, if the milk get decolorized, so the milk is considered unsatisfactory, means the test is positive, means it shows that yes, bacteria is present. But if the milk is not decolorized within 30 minutes, the milk is considered as the good quality. So a positive result occur when the viable, means bacteria are present in the milk, it decolorizes the milk within a certain period of time, like 30 minutes. And if within 30 minutes if the milk does not decolorize, so the test is negative. So if the reduction occurs within 30 minutes, means this is to be noted very, you have to understood this thing very properly, that if the reduction, means if the color is reducing within 30 minutes, then the quality of the milk is very poor. If the reduction is occurring between 30 minutes to 2 hours, then the quality of the milk is still poor, not very poor, it's poor. But when the reduction occur between 2 hours to 6 hours, then the milk then the quality of the milk is quite fair. And if the reduction occurs between 6 hours to 8 hours, then its quality is good. And finally if not reduced in 8 hours, then obviously the quality of the milk is excellent. So that I have shown this in the this type of table. So this shows the grading of the milk in NBR test. So when the quality of the milk is good, I am just marking here, you can note it down somewhere. So when it is good, means after within 5 hours, and means if the reduction time is 5 hours or more, means approximately 40 hours, so the quality of the milk is set or good, and the bacterial count is below 50,000. Then if the quality of the milk is fair, when the milk is fair, between 2 to 5 hours, like 30 minute approx, 30 minutes, sorry, 30 hours, then the bacterial count would be itna. When it is very bad, within 20 minutes to 2 hours, like 10 hours, means jitni jaldi, agar if in Hindi I will tell you, jitni jaldi color reduce कर देगा उतनी ही ज़्यादा milk की quality is poor. Okay, so you must be aware of these things. So how it is added? So addition of blue in a raw milk and rapid decoloration shows like this, means this is a test tube where you are adding the methylene blue in the raw milk. So this rapid decoloration shows the milk is of poor quality. Rapid means very fast. Slow means this is more, this is more. If you talk about the quality, the contamination, so in this it is still it is very poor, it is poor, and this shows it is good. Okay, so this is how the reduction time play an important role, means within कितनी जल्दी color decolorized हो रहा है. Now moving to the next, that is what are the factors that affect the MBRT test. So the first factor is the rate of decoloration depends on the type of organism. Okay, means the rate of reaction depends upon the type of organism. For example, the coliform bacteria are the most rapidly reducing organism, and thermodiuric and psychrotropic bacteria, it reduces the methylene blue very slowly. So the rate of reduction, so the first factor is the rate of reduction, so it depends upon the type of microorganism. Second is the reduction time of mastitic milk that will affect the presence of large number of leukocyte. Then third, light hesitance reduction processes, and therefore MBR test should be carried out in relatively low light. The fourth factor says the time of reduction, it depends on the concentration of methylene blue solution. The concentration should be uniform, otherwise, means the concentration should be uniform, otherwise the increased concentration of dye can increase the time of reduction. Then the last factor says the activity of bacteria in the milk sample. So the activity of bacteria in the milk sample is increased with the increase in the incubation temperature, and therefore shorten the reduction time. So these are the factors that affect the MBR test. Now what are the advantages and disadvantages of this test? So MBR test, if we talk about the advantages, so advantages says it the test is very simple and the test is very rapid. It is inexpensive test, only a viable cell activity reduce the dye. What are the disadvantages? So if we talk about the disadvantages, so not all the bacteria reduce the dye equally, means every bacteria have their way of reducing the dye. So depending upon the type of bacteria, the dye get reduced, so not every bacteria reduces the dye equally. So this test is not applicable to food containing reducing enzyme. So these are certain advantages and disadvantages of the MBRT test. Now further moving to the some frequently asked question in the interviews as well in the examination. So before that, let us talk about the conclusion of today's experiment. So the conclusion says that methylene blue reduction test, it gave an accurate result about the quality of the milk and the amount of contamination contaminant, or you can say microorganisms that are present in the milk. So what are the frequently asked question? So the first question is, what is MBRT test in the milk? So in exam, in interviews, this question is always there. So what is the MBRT test? So MBRT test, that is methylene blue dye reduction test. It is commonly known as MBRT test, and it is used as a quick method to assess the microbiological quality of a raw as well as the pasteurized milk. Okay, so this is the answer for the question. Now the second question says, does methylene blue kill bacteria? Means methylene blue have the capacity to kill the bacteria. So methylene blue is used less frequently as it is highly toxic to plants and will wipe out bacteria in a biofilter. Now the third question says, what is the application of methylene blue dye reduction test? So the methylene blue dye reduction test determines the growth and death rate of microorganism. And lastly, what is the function of methylene blue? So methylene blue, it is used to stain animal cell to make their nuclei more absorbable. It is a component of staining media such as red stain and Jenner stains. Methylene blue is also known as methyl theaninium chloride and is a photoactive phenothianese dye. So these are certain questions that can be asked in the interview. So you should you just take a screenshot of this slide, and you should be aware of these questions. So that brings to the end of the lecture. I hope you have understood the lecture. If you have any doubt, any query related to the lecture, you can simply leave your queries in the comment section below. In the next lecture, I will be answering the query. You can follow ITLS Academy on Twitter, Instagram, Facebook, and this is our website, that is www.itlsacademy.com. You can simply visit this website and get updated. Thank you for watching. Please do like, share and subscribe. Thank you.